Well, this is it. This is the last Biochem video. Um, yeah, I'm kind of bummed out. I miss talking about biochemistry. I'm gonna miss it. I am done tutoring after this, and originally I was like, oh man, I'm kind of busy. I don't know if I'm gonna put one out, but I figured I made it this far. I might as well make a final video for the final stretch and all the information in this video. Some of it will be review because we're a little bit ahead of where I'm starting since I haven't put a video out in a little bit, but everything I'm going to cover today is going to get you all the way to the final exam. And towards the end, there is another packet called the Teresa Porter packet that kind of covers the last bit of information that you are going to need for your final, not necessarily exam three. And when I took exam three last try, it was one of the hardest biochem exams I've taken. So I'm going to try and do the best job that I can. And honestly, towards the end, I am not an expert on the information at the end. So if you think you're sitting there like, what is this guy talking about? What's he saying? Odds are, I really don't know. I'm just kind of reading off the packet and interpreting it in my own way. So at this point, if you guys want to turn it on 2 times speed and get flowing through this, because I'm going to be talking for a while. This is probably going to be, if I can keep it under an hour and a half, I'll be happy. So yeah, turn it on 2 times speed and get cooking. So we'll start off with gluconeogenesis precursors. So we know gluconeogenesis, basically main regulation to take glucose precursors from our body, muscle, liver. Actually, gluconeogenesis doesn't occur in muscle, but we'll kind of get into the inner workings of that later on. But basically, gluconeogenesis is getting glucose back into our blood to regulate our blood sugar through influences by insulin, glucagon, epinephrine, which we'll be covering in a little bit. So precursors: lactate, glycerol, and amino acids. So lactate and glycerol, they can go straight into gluconeogenesis. They can go right back up the path. They can be eventually turned into precursors to get to like oxaloacetate and eventually just go back up through gluconeogenesis, or they can just kind of go right back up the basically just the reversal of glycolysis. But we've covered gluconeogenesis so much already that honestly I feel like everyone kind of knows that. And then amino acids. So amino acids, which we will get into as well, they can just pop into different points on the TCA cycle. They can go through transamination reactions, or they can just pop right in. Like some amino acids go to like fumarate. You can see some of the amino acid symbols. Oh no, is my pencil not working? Oh, whew. You can see some of the amino acids just popping in right there, like alpha ketoglutarate, succinyl CoA, fumarate. So these amino acids, for the most part, are just going to be popping in on the TCA cycle to help promote gluconeogenesis. Now, fatty acids cannot be a part of gluconeogenesis because realistically, when fatty acids are converted into acetyl-CoA, because a lot of fatty acids within inside our body are going to be turned into ketone bodies, which will go through the whole ketone body formation, everything like that. These really cannot be used for gluconeogenesis because they did not come from the PDH system. And the PDH system, if they did not come from it, it cannot be reversed through that system. Only acetyl-CoA that comes from the PDH system can be precursors. So if you guys remember the pyruvate dehydrogenase system, to where we went through glycolysis, we get to pyruvate. Pyruvate is acted on by pyruvate dehydrogenase to produce acetyl-CoAs. These acetyl-CoAs can be used for gluconeogenesis, but the acetyl-CoAs generated from fatty acids cannot be used for gluconeogenesis because most of those acetyl-CoAs are going to go towards ketone body formation. Little exception too that I'm reading right here. Exception is propanol-CoA. It's a product of beta oxidation and it has an odd number of saturated fatty acids. Do I know the inner workings of that? Not really, but it's a fun fact. So keep that in your brains. Ketone body formation. Yeah. So when all of oxaloacetate has been used up during gluconeogenesis and aerobic respiration, acetyl-CoA accumulates. And this accumulation is basically because if we remember to start the TCA cycle, we need acetyl-CoA to combine with oxaloacetate to produce citrate. So if we are using oxaloacetate for gluconeogenesis, we do not want to take that same oxaloacetate and combine it with these leftover acetyl-CoAs because we're just taking away our components of gluconeogenesis. So our body, when in gluconeogenesis, we like to keep oxaloacetate specifically in gluconeogenesis, and we will take those overaccumulation of acetyl-CoAs and put them towards ketone body formation. And I can't even remember, do I have to talk about it down here? No, no, no. We already covered ketone body formation, I'm pretty sure. Uh-oh. Yep, no, we did. Okay. Yeah, so go back and review that in the later videos. I am not going to talk about that again because I hate ketone bodies with a passion. So, where was I? Lack of oxaloacetate means we can't use the acetyl-CoAs, da 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 da. Alternate pathway. So, what basically we're going to be talking about pyruvate carboxylase, and what pyruvate carboxylase is we are converting pyruvate straight into oxaloacetate, and this mechanism is caused by the overaccumulation here of oxalo or acetyl-CoA. So when we have that overaccumulation right in this area, right there, it's going to say, hey, pyruvate, we don't need you to turn into any more acetyl-CoAs. We already got enough down here. We're already not being used. And so what it's going to do is it's going to go boop, boop, shut off that pathway right there. So we can't use the PDH system anymore. There's no need. We already have too many acetyl-CoAs that we need to put towards ketone bodies. So instead, pyruvate is going to go, crap, where do I go from here? And this is where pyruvate carboxylase comes into play. Thank goodness I was not a writer or anything because my handwriting is horrible. So basically what's going to happen is when acetyl-CoA shuts down this pathway right here, pyruvate is going to go straight from here. Pyruvate's going to turn right into oxaloacetate through the enzyme pyruvate carboxylase. And pyruvate carboxylase kind of stands for is, hey, we're acting on pyruvate. And if we look right here, we can see we're throwing on a CO2 right there. So we're adding a carbon on, and we're turning it into oxaloacetate through pyruvate carboxylase. So pyruvate gets turned into oxaloacetate, and we can proceed with gluconeogenesis from there when we have that overaccumulation of acetyl-CoAs that are not being used. This is just kind of an alternate pathway to keep gluconeogenesis going, and it also inhibits the PDH system, which again, we just talked about that right there, because why would we use the PDH system to turn pyruvate into acetyl-CoA when we already have too many acetyl-CoAs, So yeah, that's a little ramble right there. So now, honestly, I'm having fun with this. This is the last one. I'm feeling kind of giddy, kind of done doing. I'm ready. Enjoyed tutoring, but moving on to other things now. Hormone effects on blood glucose. All right. So here's what we're going to be talking about. Hormones, I guess. So main hormones we're going to need to pay attention to are insulin, glucagon, and epinephrine. And we also need to know the types, where they come from, and the stimulus, what causes them to be released and be created. And then furthermore, what do they do to blood glucose? So let's talk about it. Insulin is a polypeptide. It is created in the pancreas, and the stimulus to create insulin is when we have high blood sugar. So if you're just down in those really good sugar cookies that you can get from Walmart, with like the nice frosting on top, you're just going to eat a ton of those, and your blood sugar is going to spike, and so your pancreas is going to be just producing insulin. What insulin does is it senses all that sugar, that high blood glucose levels or whatever in your blood, and when insulin acts upon it, it allows our body to take that glucose out of our blood and put it into peripheral tissues to lower our blood sugar. Now, on the contrary, we got glucagon. Glucagon, again, is another polypeptide. It's produced in the pancreas, and the response of glucagon is basically it senses when there's low blood sugar. So this is where gluconeogenesis comes into play. And as you're probably guessing, glucagon helps promote gluconeogenesis. So when your blood sugar just drops dramatically, you don't have any of those fruit snacks or any of those cookies in a while, your pancreas is going to go, hey, glucagon, start kicking on gluconeogenesis to get some blood or get some glucose from our peripheral tissue to put back into our blood. So when glucagon's released, we're snagging all this extra glucose that we put into all these storages, we're releasing it and putting it back into our blood. And what's that going to do? Oh, it's going to increase our blood glucose levels. And then we have epinephrine. Epinephrine is the odd one out of the bunch. It's in fight or flight, and it actually has two different effects. It can affect our liver, and it can affect our skeletal muscle. So there is going to be a little differentiation we need to make there in a little bit. But epinephrine, catecholamine, it's created in the adrenal medulla. Stimulus is fight or flight, and basically the effect on blood glucose is it helps raise our blood glucose. Little fun fact right here that we need to know. Glucagon receptors in the liver only, insulin receptors both in skeletal muscle and adipose tissue, and then epinephrine receptors both muscle and liver. lock that into your brain's free bit of knowledge right there. All right. So we're going to be getting in and talking about CAMP, or CAMP. And this molecule is really needed for when glucagon's activated. And basically, this is a precursor for this huge cascade of reactions that we're going to get into in a little bit. But we should probably just talk about how it's made. So, CAMP is made from cleaving inorganic pyrophosphate from ATP. And basically, what I want you guys to know is how camp is going to be made. And I know, like, we're already in week like 12 or 13, and you guys have already taken a quiz on this, so I'm sure you guys are just flying by and not even listening to this at all right now. C A M P A T P, and then we'll have this little enzyme called adenyl. I spelled that wrong. Whatever, you guys get what I'm trying to say. Cyclase. So basically what's going to happen is ATP is going to come in the intracellular component. It's going to react with adenyl cyclase, and we are going to produce CAMP, and we are also going to produce PPI. Lock PPI into your brain, because just like the creatine examples from the last exam, he's going to be like, hey, what are the products when we use adenyl cyclase on ATP? And we're going to go, oh, well, our products are PPI and CAMP. Pretty simple. So, as I just said, enzyme involved, adenyl cyclase, helps cleave that ATP and produce CAMP and PPI. CAMP is a secondary messenger of glucagon, and it helps activate protein kinase A. And protein kinase A, you guys will also see it as PKA. When you guys are rocking out to those practice questions, PKA is going to be everywhere, so do not get confused, because you're here to hear first. So let's talk about glycogenesis, production of glycogen. Glycogenesis, basically it's glycogen synthase or synthesis, and it uses the enzyme glycogen synthase. So this is basically, if you guys remember all the way back to glycolysis, we'll have glucose here, G6P, da 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 da. Oh crap, I'm gonna have to change colors. So basically what's happening is G6P is going to turn into G1P, and then we're going to turn down here into glycogen, and then we have some reactions back. Glycogen synthase is going to be right here in between G1P to glycogen, because as we can tell, glycogen synthase, we're making some glycogen, so it's going to turn G1P into glycogen. Then we have glycogenolysis, which is basically the breakdown of glycogen, and that's just going the other way. So the other way being right here, because I don't know if you guys are keeping up. So glycogen phosphatase, we're going to have a phosphatase enzyme right here, and basically what a phosphatase enzyme will do is it will add on a phosphate group right here onto glycogen, turn it back into G1P. G1P can go back up into G6P, and it can either, if it's in the liver, we can go back to glucose, or if it's in skeletal muscle, we can put it down glycolysis and have it be created for energy. And also too, both of these, for the most part, are mainly accomplished in the liver. We do have this technically, the forward motion of glycogen G1P back to G6P can happen in skeletal muscle when we need to release it, and we'll get into that with epinephrine, when we need to release it to make energy for our muscles when, like, let's say you're running away from a bear when you're out on one of those great hikes in Missouri. Missouri does not have really any good hikes. Like, the forests around here kind of stink. It's farmland. Like, no good lakes either. Like, bring back Lake Michigan, man. We need one of the Great Lakes down here. So, function of glucagon. Okay, here we go. Glucagon. What do we remember about glucagon? It's a polypeptide produced by the pancreas. It's in response to low blood sugar, and it helps initiate gluconeogenesis, which means we are taking glucose-style components and helping put it back into our blood to raise our blood sugar. So what we do need to know is glucagon is always an extracellular component. So it's going to bind to the outside of the cell right here, and it's going to initiate a cascade on the inside, or the intracellular aspect of the cell. So what glucagon is going to do, it's going to activate a G protein, which in turn promotes the activity of adenyl cyclase. And we just talked about adenyl cyclase cleaving ATP to produce CAMP and PPI. So how this is going to go, glucagon is going to bind extracellularly. Bleh. Right there, that G protein is going to come down over here. ATP is going to come in, react with adenyl cyclase, which we can see by this little E representing an enzyme. ATP is going to come up, react with adenyl cyclase and that G protein, and then boom, we produce CAMP and we produce PPI. So now what is CAMP going to do? Well, CAMP is going to basically activate protein kinase A, which here's the inactive form of protein kinase A, and look right here with all these camps bounded on there, that's the active form of protein kinase A. And protein kinase A is going to have a ton of functions. So basically what it's going to do is CAMP is going to activate phosphorylase kinase A, which is right here. So protein kinase A comes down here, activates phosphorylase kinase A. Phosphorylase kinase A is then going to activate phosphorylase A, which is right here. And then phosphorylase A, what basically that's going to do is it's going to allow for glycogen to be released and turned back into G1P, which we just kind of talked about up here, back into G1P. G1P can go back up into G6P and then be turned into glucose, and that can be released into our blood. Yeah, and what it's also going to do is it's going to inactivate glycogen synthase. So how this mechanism works is as soon as glucagon activates and it activates protein kinase A is going to be promoting this side to release glycogen back into G1P, but it's going to be inhibiting this side over here because if we're creating more G1P, we don't want glycogen synthase activating to take that G1P and turn it right back into glycogen. That would just be counterintuitive, and it just would not be fun. And realistically, we just be, I don't even know the effects of what happened at that malfunction. Like, you're just in a constant glycogen cycle. I don't know. Really don't know what that does. Fun fact right here, glucose is transported to the blood plasma via transport protein GLUT2. Phosphorylase kinase A can be activated by binding of calcium to calmodulin unit of the enzyme. We'll talk about this in a second. We don't need to know calmodulin complex for glucagon. Glucagon's half-life, 10 to 12 minutes. Big enzyme to know here. So we know CAMP activates protein kinase A basically right there, and we activate this whole huge cascade down here. But when we are done promoting gluconeogenesis and we want protein kinase A to become activated, we use the enzyme phosphodiesterase. Yeah, phosphodiesterase. We use the enzyme phosphodiesterase to remove CAMP from protein kinase A right here, so it turns back into its inactive form. So when we're done, we're like, hey, shut off gluconeogenesis. We throw phosphodiesterase at it, and we say, all right, there we go. CAMP removed, protein kinase A inactivated. Now, it also has a third function because you saw protein kinase A has three functions, and we only talked about two. So here's the last one. So protein kinase A activates fructose 2,6-phosphatases and inhibits PFK2. And this step costs 1 ATP, and activation of fructose 2,6-phosphatase causes the hydrolysis of fructose 2,6-biphosphate, blah, 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 blah. I'll show you guys the graph and we'll walk through it because those words just starting to confuse me even. So basically, we talked about this in the last video, and if you guys were at a couple of tutoring sessions a couple weeks ago, we kind of went more in depth about this. So basically what's going on is we already talked about this. The more fructose 2,6-biphosphate we have, we're promoting glycolysis. And the more fructose 2,6-biphosphate we have, the reason it's in glycolysis is because we are a positive modulator for PFK1. And if we remember, PFK1 helps the conversion of fructose 2,6 or fructose 6-phosphate into fructose 1,6-biphosphate. And it's also a negative modulator for fructose 1,6-biphosphatase, which would turn fructose 1,6-biphosphate back into fructose 6-phosphate. So what glucagon, the third function of glucagon is doing is activating this enzyme right here, fructose 2,6-biphosphatase. And what this is doing is it is converting fructose 2,6-biphosphate back into fructose 6P, and we are lowering our storages or our number of fructose 2,6-biphosphate molecules. Now, when we lower that amount, we can go and see right here, less fructose 2,6-biphosphatase promotes gluconeogenesis. And this is because when we have low levels here, we reverse all of our inhibitors basically. So when we had a high amount, we were a positive modulator for PFK, but when we have a low amount, we are a negative modulator for PFK1, which means we inhibit PFK1 and prevent glycolysis. And if we zoom around the racetrack right here, all the way over, we can see fructose 2,6-biphosphate or phosphatase, whatever it is. We're a negative modulator for fructose 1,6-biphosphatase. But now when we have low levels, this enzyme gets kicked on right here. And fructose 1,6-biphosphatase is now converting fructose 1,6-biphosphate back into fructose 6P, and we're putting it down the path of gluconeogenesis because we are going back up the steps back to create glucose. So that is how glucagon acts upon fructose 2,6-biphosphatate and influences gluconeogenesis upstream. When we have low F2,6-biphosphate levels, gluconeogenesis. When we have high fructose 2,6-biphosphate levels, we are going through glycolysis. All right. Talked about that. Only occurs in hepatocytes because we know gluconeogenesis can only occur in hepatocytes. So now let's talk about epinephrine and glycogenolysis. Sorry, I had to take a sip of coffee there. Yeah. So big receptors we got to know here. Epinephrine can bind to different cells. We can bind to muscle cells or we can bind to liver cells. So in the liver, we are going to be binding to adrenergic receptors. And in the muscle, we are going to be binding to beta receptors. And we kind of noticed that in the liver, epinephrine had a similar function as glucagon, which promoting gluconeogenesis. So when we bind to the liver, when it binds to whatever that word is, receptor in the liver, we're going to be promoting gluconeogenesis. And so here, epinephrine again is a extracellular component binding to this receptor right here. We again release this G protein, and a couple of these reactions right here, honestly, don't even worry about them. Don't even worry about some of this stuff right here. What we really want to worry about is this little gate opening right here to allow an influx of calcium ions. And we just talked about calcium, and I said, whoa, hold on, we're not ready to talk about that yet. Well, now we're ready to talk about it. So basically, we're going to be activating this thing called the calmodulin complex. And really what you guys need to know, epinephrine, boom, binds to this receptor, releases this G protein. And then as a result of all this fluff right here, this channel gets open and allows an influx of calcium. This influx of calcium is going to activate this calcium calmodulin complex. This calmodulin complex is then going to activate phosphorylase kinase. And phosphorylase kinase upstream is just going to allow for the release of glycogen back into G1P, back into G6P, back into glucose. Really quick, I'm kind of flying through it, but a lot of the steps are very similar to glycolysis. It's just when you think about epinephrine, you want to be thinking more about the calmodulin complex. When you're thinking about glucagon, you're basically going to want to be thinking of all these like protein kinase, phosphorylase kinase, stuff like that. Now, epinephrine in the muscle. We use a beta receptor, and we can use both a calmodulin complex and we can use CAMP when epinephrine is acting in the muscle. So epinephrine, boom, binds to a beta receptor. This G protein is released, and we can see we have adenyl cyclase again. Adenyl cyclase, what we know is it helps cleave an ATP to produce CAMP. CAMP goes down its huge cascade, and we can see right here PKA, protein kinase A, and protein kinase A will eventually act upon phosphorylase kinase A, but we can also see we can have an influence of calcium here through a nerve impulse. This nerve impulse will help open up the calcium channels to help release calcium back into the cell and activate the calmodulin complex. So we can, in muscle cells, we can have both a nerve impulse and an epinephrine response as well, both happening simultaneously. So a little bit different here. So in the past two steps that we talked about, we're talking about gluconeogenesis with the conversion, or taking glucose and putting it back into a blood. When it's in muscle cells, we're trying to give the muscle cells energy, and we know in muscle cells gluconeogenesis cannot occur. So when epinephrine is happening or occurring in the muscles, we are kind of promoting glycolysis because we're trying to release glycogen storages from our muscle and allow these glycogens to be made back into G6P and have the G6Ps go through glycolysis, start pumping out ATP to start giving us energy for our muscles. So when we're running away from a bear or something like that, you don't run out of energy, because that would really suck if you're just like half a mile away from the bear and you're just hauling butt and that thing's catching up on you, and then boop, no energy, and all your muscles fail and you sit there. I mean, some bears, you can, if you lay dead, they won't attack you, so it might work out for you, but I'm not willing to take that risk. I'm gonna make sure I got some epinephrine in me. Oh man, I just got on a tangent right there. I apologize. Yeah. So yeah, epinephrine basically just increases fuel for the muscle. Little fun fact stuff right here. GLUT2 transports glucose from blood to liver kidney, and intestines. It is insulin independent, and then GLUT4 transports glucose from blood to muscle, skeletal, and cardiac, and adipocytes, and it is insulin dependent. Know the difference between these two channels right there, because they are just probably going to be word bank questions. All this stuff right here, you guys can just read through. We've been hammering gluconeogenesis like crazy, so I'll leave that up to you guys. Here is just talking about glucagon and insulin levels during the day. So if we look right here, we can see at 6 a.m., I don't know who's eating at 6 a.m. If you are, you're a madman. But if you eat at 6 a.m., you can see you have a spike in insulin, because insulin is acting to take all of this blood glucose that we've been ingesting and take it out of our blood and to put it into peripheral tissues. And we can see also when we eat, we have a drop in glucagon levels, because we don't need to put any more glucose into the blood if we already have a high blood glucose level. And then you can see in areas where insulin is inactive, that's when glucagon comes back up to maintain that regulation, to make sure we have a constant equilibrium between blood glucose and make sure nothing's dropping too quickly, and making sure we're not passing out. Yeah, I'll let you guys read through this. All right. Now we're kind of cooking. We only got 12 more pages. Well, actually, probably 14 more pages to go. All right, let's keep on cooking. So fatty acid biosynthesis. Yeah, this is where we're making fatty acids, adding chains onto fatty acids, and you guys are probably looking at this like, holy cow, what's going on? Well, I'll tell you. So basically, name of the game, add carbons onto a fatty acid to increase its chain length. And anytime you see a CoA, you're kind of assuming fatty acid. So we can see we have acetyl-CoA right here. We'll have a malonyl-CoA right there, and we'll get into what those do. But we just need to know about some of these steps in here. So acetyl-CoA carboxylase, we can see that is inactive right here in its normal state. And when it's acted upon by B7 and protein phosphatase, which, look, right here, we can see we're removing a phosphate. And actually, this is a big concept we're going to get into in a little bit with cholesterol synthesis. Most of the time when we have a lot of inactive enzymes, it's because we throw a phosphate group onto there. So protein phosphatase, what that's doing is acetyl-CoA carboxylase right here probably has a phosphate group that latched onto it. So it is remaining inactive. So when we throw B7 on there and also act upon protein phosphatase, we can remove that phosphate group and turn acetyl-CoA carboxylase into its active form. Now, once that is in its active form, we can take this enzyme and throw it over here. Now we have acetyl-CoA carboxylase active in parentheses, so we know it is active. So basically what this enzyme is going to do is we're going to take acetyl-CoA. Again, we're going to use vitamin B7 here, lock that into your brain for fatty acid biosynthesis. And then we can see right here, we use an ATP to throw a carbon on. You can also use HCO3, but for here, we're just going to use a CO2. So we're going to throw a carbon onto acetyl-CoA and turn it into malonyl-CoA, which is a 3-carbon structure. And again, we need biotin B7 to allow this whole thing to work. So now we've talked about activating acetyl-CoA carboxylase, which acetyl-CoA carboxylase is going to throw a carbon onto acetyl-CoA with the help of B7 and ATP. We're going to be producing a malonyl-CoA, which is a 3-carbon structure. And we can get down here. We can see that we have a fatty acid, which is already just a 4-chain fatty acid. And let's say we want to add carbons onto that fatty acid structure. We can use malonyl-CoA, which is a 3-carbon structure. And hold up, what's going on there? We have a fatty acid with only 6 carbons. Where'd the other carbon go? Well, if you guys look right here, we get rid of a CO2 group. So technically, we add malonyl-CoA onto this fatty acid chain, which is a 3-carbon structure, but we get rid of a CO2 in the process. So realistically, we're only adding 2 carbons on at a time when we're adding on to fatty acid chains. So keep that in your mind. When you're building a fatty acid chain, we're adding on 2 carbons. And we've talked about breaking down fatty acids and stuff before with beta oxidation. And you guys know we cleave 2 carbons off at the same time. Same exact concept here. Add 2, take away 2, whatever he's talking about. All right, let's talk about A, B, and C here. A, protein phosphatase right here, is stimulated by citrate and inhibited by long-chain fatty acids and acyl-CoAs. So Yeah. Let's say we have just an overabundance of citrate or something like that, and we don't need to make any more citrate. Well, citrate, an overabundance of citrate is going to activate protein phosphatase A, so we can take those leftover acetyl-CoAs and turn them into fatty acid chains. But it is inhibited by long chain fatty acyl-CoAs, which basically saying like, hey, if we have a ton of fatty acids with a ton of long chains, we really don't need to be adding to them. So now we look at B, which acetyl-CoA carboxylase active, it's stimulated by insulin and inhibited by epinephrine and glucagon. And basically what this is just saying is with insulin, we know we're trying to get glucose out of the blood and everything like that. And this insulin kind of helps get glucose out of the blood and put it through a couple different processes for storage. And fatty acids is just going to be one of our main storage components. And we'll kind of get into cholesterol and triglycerol storage through chylomicrons and everything like that in a little bit. But insulin is basically just promoting the synthesis of fatty acids by activating acetyl carboxylase. And it's inhibited by epinephrine and glucagon because, again, we're taking components, trying to put it back into the blood. It's not trying to create any structures. We're trying to modify our blood glucose levels. And C, AMP dependent kinase, this little enzyme right here. All it's going to do is just take ATP and throw a phosphate group back onto acetyl-CoA right here, or acetyl-CoA carboxylase, and turn it back into its active form. And it's going to be activated by AMP, glucagon, and epinephrine because, again, if we are trying to put stuff back into the blood, we do not want to use those components we need to make any larger structures. So it's going to be over here saying, hey, acetyl-CoA carboxylase, we don't need you anymore, man. We need to regulate our blood sugar. And it's going to throw that phosphate group on there and turn it off. Yeah. Memorize these steps right here. This will just be probably a quick question for you guys. He's probably going to ask about the order, like, oh, what happens? What happens for fatty acid biosynthesis? Well, number one, decarboxylation, removal of CO2 group, reduction of the substrate via NADPH oxidation, right here, dehydration, removal of H2O, right there, the reduction of the substrate via NADPH oxidation. Something in there. There's, I don't know. Just memorize these. I know that much. NADPH is supplied by pentose pathway, which we talked about that a long time ago. So yeah, pentose pathway produces NADPHs for the building of structures like these right here. We knew it was part of the anabolic, very anabolic style processes, and this is kind of where it's all coming back into play. Oh, all right. Here's basically just going over what we just talked about. Study it, be good at it. The number of cycles is equal to 1 less than half the carbon length intended. 16 requires 7 cycles. We kind of talked about this with the beta oxidation cycles that we talked about. So now we're going to talk about the pyruvate citrate cycle. And I'll be completely honest with you here. I still don't completely understand this. My brain must have shut off or something like that. And even last try in tutoring, man, I mean, this was a pain in the butt for me to get down. So we're just going to walk through it. I'm going to kind of explain some of the key features we need to know. But when it comes to some of these products and like the creation of another pyruvate and stuff right here, I really can't get that much in depth with it. So I hope you guys have been going to Annalise because I know she is excellent at what she does, and she probably has a way better idea on how to do this than me. So, pyruvate citrate cycle. What we need to know is 2 pyruvates are going to enter the mitochondrial matrix, and each of them are going to go down a separate pathway. So we can see glucose 6 carbon right here, gets turned into two pyruvates, and each pyruvate is a 3 carbon structure each, so 2 times 3, we still have 6 carbons right through the mitochondrial matrix. Basically what we need to know is we're just trying to get acetyl-CoA products here. We're just trying to get acetyl-CoA for fatty acid biosynthesis, but acetyl-CoA cannot cross the mitochondrial membrane, and we really can't extract acetyl-CoA out here in the cytosol right away. So we need to throw pyruvate into this mitochondrial matrix, and let's talk through some of this stuff real quick. Pyruvate right here, both of them are going to go on separate pathways. We are going to go through gluconeogenesis, or kind of pyruvate carboxylase, style reaction right here, because remember with pyruvate carboxylase, we turn pyruvate into oxaloacetate with the addition of a CO2 right here. And we're basically saying, hey, pyruvate, we're going to throw a carbon onto you, turn you into a 4-carbon structure for a quick minute to turn you into oxaloacetate. And pyruvate's going to be like, hey, all right. And we need B7 right here as well. Don't forget about that. And then we're going to take the other pyruvate and say, hey, no, you're going through pyruvate dehydrogenase system, and we're going to remove a CO2 right here. So we create a 4-carbon structure with oxaloacetate over here, and we create a 2-carbon structure with acetyl-CoA over here. But we still have a total of 6 carbons. So we can see PDH system right there, pyruvate carboxylase right here. We'll jump down here, and we have oxaloacetate and acetyl-CoA. And what they are going to do, they're going to turn into citrate because, the TCA cycle occurs in the mitochondria. So this allows for the combination of oxaloacetate and acetyl-CoA to create citrate. Citrate is going to fly back out of the mitochondrial matrix right there. And we're just going to go through a couple reactions right here. Really can't explain them, but citrate lyase acts upon citrate. And basically citrate lyase, what it's going to do is it's just going to revert citrate back into its 2 separate components, oxaloacetate and acetyl-CoA. And kind of zooming back out here, acetyl-CoA, what I really just want you guys to know, this acetyl-CoA is going to be made into palmate, which is a final fatty acid product. But this oxaloacetate over here is just going to go through malate dehydrogenase and some reactions right here. And oh crap, sorry guys. Oxaloacetate first is going to have a hydrogen thrown on here from NADH. And if we remember, I don't want to waste too much time going over it again, but basically when malate is turned into oxaloacetate in the TCA cycle, we are dehydrogenating malate, or we are removing, we are removing a hydrogen to turn it back into oxaloacetate. So in this instance, we want to turn oxaloacetate back into malate. So it can kind of go and be turned back into pyruvate through some gluconeogenesis style pathways. I guess there was a chart earlier that I believe I talked about in the last video that covered this, but basically oxaloacetate gets a hydrogen thrown back onto it. It turns into malate. Malate will then be acted upon by malate dehydrogenase, and we will produce a pyruvate to go back up and begin the cycle again right there. When carbon is lost, one of the 2 pyruvates is recycled back into pyruvate again. So like we have 2 pyruvates basically coming out all the way to this point. One of the pyruvates is going to be made into palmate, and the other pyruvate is just going to be recycled back up there through all these crazy different reactions. For the finer details of the system, please go through the practice problems because I'm probably butchering the explanation of this, but I'm just here just kind of walk you guys through it. But yeah. There's a lot of stuff for this, but no worries. We're just going to keep on crack-a-lacking. Malate shuttle and pentose pathway are responsible for replenishing the levels of NADPH so that the reducing agent can continue to reduce malonyl-CoA to the FA palmate or fatty acid palmate. And this is kind of the same system right here. We're talking about NADHs and everything like that right there. Oh, carbohydrates are the main source of fatty acid biosynthesis, which kind of makes sense. We eat a ton of carbohydrates. Insulin's trying to rush to take it out of our blood. We're activating acetyl-CoA carboxylase to convert these acetyl-CoAs into malonyl-CoAs. These malonyl-CoAs will go and react with the fatty acid chain. They'll go through the reactions with NADPH. It's going to remove a CO2, going to remove an H2O, and we're going to basically just add 2 carbons onto these fatty acids to make these longer fatty acid chains. Cool. Okay. Lipid synthesis. Now we're kind of getting into, I guess, a little bit more of the fun stuff. We're crack-a-lacking. We're only 10 pages away. So lipid synthesis. Lipids are made to store energy when the supply of energy metabolites, glucose and glycerol, exceeds demand. So this is kind of where DHAP comes into play. If we remember DHAP being kind of that secondary role in glycolysis that we had. So if we have too much energy and everything, it's going to send it down to DHAP, and DHAP is going to be making it into basically lipids and storing it in adipocytes. So in the liver, both glucose and glycerol are precursors to lipid synthesis. Liver equals glucose and glycerol. And lipids are going to be made only in hepatocytes from glucose, but lipids can be made from glycerol and glucose in the liver. And then the fats we're going to be talking about, or the lipids we're going to be talking about here, are triglycerols. So insulin is used. Insulin is always anabolic. Um, Insulin anabolic, meaning we are creating structures, and then we can see glucagon and epinephrine right here are always catabolic, which means we are breaking structures down. So I know I went over this in the last tutoring session. We're finally kind of catching back up to where all of the kind of catching back up to where I was, because I did end my last tutoring session yesterday, and I know we finished it all. So now we're kind of catching back up to being on track, and hopefully you guys watch this before your next quiz. I do have a draft for the NFL or fantasy league in a couple hours, so hopefully I can get this thing uploaded and sent out to you guys before I have to leave for that. But we'll talk about desaturation of fatty acids. So basically, desaturation of fatty acids, what we really need to know here is just another little math thing, and it's kind of like a puzzle. So all we need to know is when we desaturate a fatty acid, we're just adding a double bond to a saturated fatty acid. So like if we have a 16-0 fatty acid right here and we put it through desaturation, we're basically turning into 16-1 delta 9. And like if we want to, and realistically, we can't put it through another desaturation reaction. What you're going to see in your problems is he might give you like, oh, we have a 16-0 fatty acid, but now we have, I don't know, a 20-2 delta 9 delta 12 fatty acid. What are the steps to get there? And basically, he's just going to give you a different combination of desaturation and elongation. So what's an elongation? An elongation is basically just the addition of two carbons through that fatty acid biosynthesis little step we talked about there. So 16-0, we go through a desaturation. We create a fatty acid that is 16-1, meaning that one meaning we have one double bond, and that double bond occurs at carbon number 9. Now, if we put it through an elongation, we are going to be adding two carbons to it. So we're going to turn it into an 18-1 delta 11 fatty acid right here, because when we add on these carbons, that carbon number where that double bond is going to shift, and we're going to bump it up just by up by two carbon spaces. So now, like, let's say he's like, I want an 18-2 fatty acid with delta 9 delta 11, and you're like, okay, well, we desaturated it, got it to 16-1 delta 9, and we just added two carbons onto it. So now we have an 18-1 delta 11. How do we get an 18-2 delta 9 delta 11? Well, you put it through another desaturation reaction. And all it's going to turn into is an 18-2 delta 9 delta 11. And then if you want to have some more fun and turn it into a 20-2, oh, elongation again. And you'll have a 22 delta 11 delta 13 fatty acid. That is realistically all you need to know for this. Yeah, okay, we're going to move on. Lock this whole thing into your brain right here. And now it's gone. So, sorry, it's gone. So, cholesterol. That's a biosynthetic precursor. We've talked about cholesterol quite a bit. You've probably talked about it in physiology. And it's kind of cool. So cholesterol is a biosynthetic precursor of three compounds. It's a precursor for bile salts, a precursor for vitamin D, and a precursor for steroid hormones. Steroid hormones being like estrogen, testosterone, all these different hormones that go on within inside our body. And it's also a lipid bilayer component. And I have a biology background, so I used to talk about cholesterol quite a bit with maintaining membrane fluidity within cells and helping it change states depending on the conditions that they were in. That's how it just maintains lipid bilayer component. Liver is the main site of cholesterol synthesis, but all cells can make it. But liver is the main, like it's the holy cholesterol synthesis location. Only way to eliminate cholesterol from the body is via feces or the enterohepatic cycle. So if you have too much cholesterol in the body, gone. HMG-CoA reductase is the controlling factor in the synthesis of cholesterol. So we're going to talk about, we're going to skip a lot of steps in these charts right here. But if we remember back to ketone bodies, like we needed two acetyl-CoAs. These acetyl-CoAs were acted upon by thiolase would turn them into acetyl-CoAs, and we turn it into HMG-CoA. I hope you guys remember that a little bit. So all of the steps leading up to HMG-CoA are the same as ketone body formation. But The enzyme HMG-CoA reductase is the enzyme that will turn HMG-CoA into the precursors for cholesterol. And realistically, what we need to know is we can see all these steps are pretty much, oh, red dot, there we go. All these steps are pretty much the same to get to HMG-CoA, but when HMG-CoA gets down here and it gets acted upon by the enzyme HMG-CoA reductase, it's going to turn into mevalonate, mevalonate or something like that, yeah. It's a 6-carbon structure, and this structure right here, I'll just call it M for now. M is going to go through all of these crazy reactions that I really don't need you guys to care about. But since it's written in here, we'll talk about it. There's an intermediate step right here with DPP. Basically, M right here is going to go through all these reactions, and it's just going to turn into a 5-carbon structure. Then this DPP is going to go, it's going to condense 6 times to produce 2 metabolites. We're going to create FPP and squalene. I guess so. We're producing squalene right here, and we're also producing FPP right here. But basically what I want you guys to know is it will eventually go all the way down here to cholesterol. And cholesterol is a 27-carbon structure. Wow, that's a lot of carbons. But just to recap, acetyl-CoA goes through all these same reactions, ketone body formation. We get to HMG-CoA. HMG-CoA is going to be acted upon by HMG-CoA reductase, which is going to turn it into mevalonate. Mevalonate is going to is going to come over here as a 6-carbon structure, going to go through all these reactions, go to DPP. DPP is going to come over here. We're going to create FPP. We're going to create squalene down here. Squalene is eventually going to go into cholesterol, which is 27 carbons. And cholesterol is a major inhibitor of its synthesis. So when there's too much, we're basically talking about allosteric inhibition all over again. When we have too much of a product, that product is going to come back and turn off the enzyme that's making that product, so we don't have too much and we can get back to homeostasis. And biggest thing I want you guys to know is cholesterol inhibits HMG-CoA reductase by covalent modification. Yeah, that's what, and we'll talk about covalent modification in a little bit because there's a couple other things that can inhibit cholesterol, but covalent modification is one of the main things I want you guys to know about inhibiting HMG-CoA reductase. We'll talk about some real life examples right here with cholesterol medications, aka statins, are competitive inhibitors that compete with HMG-CoA for the active site on HMG-CoA reductase because it cannot activate. So if we have a lot of statins that have very similar binding sites as HMG-CoA, these statins are going to try and clog up HMG reductase so we can't produce as much cholesterol, and we're slowing down the production of cholesterol. Yeah. Deactivation of HMG-CoA reductase uses 1-ATP. Haven't seen too many questions about that, so I just keep it in the back of your head when you're doing review. Active form of HMG reductase is dephosphorylated or dephosphorylase, inactive in its phosphorylated level. This chart right here took me so long to figure out. So long, and I think I finally cracked the code. I think I cracked it for you guys. So basically what we're going to be looking for is a positive modulator and a negative modulator. And basically these are just going to be activating or inactivating HMG-CoA reductase, so it can either create cholesterol or so it cannot create cholesterol. And basically what I want you guys to look at right here is insulin is going to be a positive modulator for HMG-CoA reductase. And it's going to stop the phosphorylation of this enzyme. And we talked about it earlier with acetyl-CoA carboxylase and how I said, oh, when it's inhibited, it usually has a phosphate group on there. Yeah, if you guys listen to that. This is what I was talking about. So I hope you guys are listening because it'll make this so much easier instead of you guys sitting there going, oh, what is he talking about? So insulin and glucagon are going to be a positive or negative modulator for the enzyme phosphatase. And we can see it in 2 different locations, here and here. So HMG-CoA in its active form is not going to have a phosphate group on there. But if we look over here, in its inactive form, we have a phosphate added onto there to keep it turned off. And one of the enzymes that's going to turn it off is called kinase P. And kinase P is just going to be sitting there with that phosphate group, just waiting for the opportunity to turn it off. And it's waiting to see if we have too much cholesterol in our body. It's going to be like, hey, let me throw a phosphate on there. Let me shut this whole system down. But insulin's going to say, hey, Get out of there. We want to keep it active. So what insulin is going to do, insulin is going to activate phosphatase, this enzyme phosphatase right here. And what phosphatase is going to do is it's going to take kinase and keep on removing that phosphate and keep kinase in its inactive form right here so it doesn't have that phosphate group on there. Because if it doesn't have that phosphate group on there, it can't throw a phosphate onto HMG-CoA reductase to turn it off. So insulin is going to make sure kinase stays as kinase and doesn't turn into kinase P to get that component to shut the enzyme off. But glucagon is going to do the exact opposite. Glucagon is going to say, hey, phosphatase, shut off. We do not want you removing that phosphate from kinase. So when glucagon is present in the system, we are going to, kinase cannot turn, or kinase P cannot turn back into kinase because this enzyme is scratched out. That enzyme is not active when glucagon is present, and kinase P will sit here and it will throw a phosphate group onto HMG-CoA reductase and turn it into its inactive form right there because it has that phosphate group attached. So just a quick recap. When insulin when insulin is present, kinase P is not here because it does not have the phosphate group, because when it's activating this enzyme phosphatase, we keep on removing the phosphate group from this kinase, and it doesn't allow HMG-CoA reductase to become inactive. But when glucagon is present, we shut off this enzyme. So like, we'll scratch all that out there. When glucagon is present, it shuts phosphatase off and allows kinase P to stay right there. So HMG-CoA reductase will be right there. Kinase P will throw on that phosphate group and turn it off. Boom. Concept one done. Now, let's say glucagon was present up here, and we turned HMG-CoA reductase into its inactive form with that phosphate group. But now we're sitting there and we're like, crap, well, we need to create some cholesterol. We have too much blood glucose, whatever, inside our body, and we need to make some cholesterol. Well, insulin is going to come in. I'm going to scratch this off so we just focus on insulin for now. Insulin is going to come in and say, hey, phosphatase. And I hope you guys notice the pattern. They act upon the enzyme phosphatase. Insulin is going to come in and turn this phosphatase enzyme on, which is going to allow HMG-CoA reductase P to react with that enzyme and remove that phosphate group right there. And HMG-CoA reductase P, as it reacts with phosphatase, that phosphate group is going to be removed and HMG-CoA reductase becomes active again. Now, let's say we don't have insulin here and we have glucagon. Glucagon is inhibiting phosphatase right here, so it does not turn on, so it does not react with HMG-CoA reductase P, so HMG-CoA reductase P remains inactive when glucagon is present because this enzyme is not activated right there, and it can't get rid of that phosphate group. So, recap, when insulin is present, we activate this enzyme, HMG-CoA reductase comes through, we get rid of that phosphate group, and HMG reductase becomes active. When glucagon is present, we inhibit this enzyme and we cannot allow HMG-CoA reductase P to fly through and become active. Holy cow, that was a lot. Another little fun thing right here is DNA can inhibit HMG-CoA reductase. Basically, I just want you guys to know, it's like we have a DNA reductase gene right here, which can I don't know, DNA can just be an inhibitor for cholesterol. I think it makes a point about it down here in a little bit. 3 forms of inhibition for cholesterol production: increased levels of cholesterol, biostals or sterols, indirect inhibition. So basically, if we have too much, if we have too much of it, it's just going to come back and say, hey, HMG reductase, we don't need you anymore. Quit making cholesterol. Statin drugs, we can eat a whole ton of statin pills as much as we want. Do not take that medical advice from me. And that's just going to be direct inhibition, because if we remember, statin drugs are competitive inhibitors for HMG reductase. So when you have statin drugs, they're going to be competing for the binding sites on HMG reductase and preventing HMG-CoA from binding, slowing down the production or stopping the production of cholesterol, which kind of sucks because we need cholesterol in our body. Like, I could go on this huge rant about cholesterol right now and how, like, big pharma, man, big pharma. So, whatever, yeah, competitive direct inhibition right there with statin drugs. And then Covalent modification. We've already talked about that. I mentioned it earlier, how covalent modification can help shut off that enzyme as well. All right, bile. Oh my goodness, we still have so much to talk about. Okay. If you guys haven't thrown it on 2 times speed by now, throw it on. Um, Bile produced in the liver via the enterohepatic cycle. It can be either recycled in the liver as insoluble biocomponents or stored in the gallbladder, released later. So basically, all we need to know, bile made here. I don't know where the gallbladder is. We'll drop the gallbladder on right there. Goes to the gallbladder, goes to the bile duct, gets released into the duodenum. Okay, big fact right here, actually, because we've talked about bile previously when it comes to carbohydrate, ingesting carbohydrates and proteins. We know that bile was used to slow down and emulsify the chyme and slow it down to allow our enzymes to act upon it and work really well to help break that down. But when it's acting upon triglycerols, we are mechanically digesting and breaking up triglycerols when bile is present in our duodenum. Whew, okay, lipid transport. Triglycerols are transported within a lipoprotein in a micelle conformation. I hope you guys remember micelles and basically amphipathic agent, Biochem 1 topic, get made into a lower energy form into micelles here, and it has a hydrophobic core, which allows us to store insoluble components inside it and transport it throughout our body. Lipids are held in the oily center of the micelle, which just talked about, and when we have lipids in the oily center, these are mixed micelles because we have the component in our hydrophobic core that we were trying to transport, and we just have an insoluble object in a soluble component. Memorize this chart right here. Personally, I never did, so take that with what you will, but we're going to be talking about chylomicrons, VLDLs, IDLs, LDLs, HDLs in a hot sec. Actually, right now, we're going to talk about it right now. And I apologize if I sound really hyper. I'm just trying to crank through this. I've had a lot of coffee today. You know, I got to Costco at 10 in the morning before everyone got there. So today's been a pretty good day so far. 2 types of lipoproteins. We have endogenous and exogenous. Endogenous sources are used to transport, or basically they're lipoproteins that are used to transport lipids made in the body. And what I want you guys to know is we use VLDLs to make that happen. We use VLDLs to transport endogenous sources of lipids. Now, with exogenous sources, exogenous lipids are sources that we consume from whatever, if you have a steak and there's a huge fatty bulb on it and you just eat it for the flavor, that's where all those dietary lipids are coming from. And what we really need to know is chylomicrons are used to transport these lipids. And we'll get into that in a second because there's a couple different steps with it, but it's a lot of fun, I promise. So we'll talk about chylomicrons. And what we know is they're used to transport exogenous sources of lipids throughout our body. This is a little bit too ahead of itself, but we're just going to walk through this really quick. So basically we're just going to look at this chart right now, and I'm going to talk through this chart and then we'll go back and reference some of the notes right there. So let's say you eat that bulb of fat. You eat that bulb of fat off of New York Strip. You paid way too much money for it, and you eat it, and you're like, I'm going to enjoy every last bite of this. Well, these triglycerols are going to get into your small intestine. Those fat lipids are going to get into your small intestines, and they're going to be absorbed or surrounded by a chylomicron. This chylomicron is going to have this triglycerol in its center. And it's not a huge detail right now, but if you guys really want to know when this nascent chylomicron for exogenous sources are in our small intestine, to begin with, we have this little thing called ApoB48 already attached to this chylomicron. Not a huge detail, nothing I think is a huge concern to know, but the next couple apos we need to know. So this chylomicron is going to kind of get out of our small intestine, be absorbed through mucosal cells, and attempt to get into our blood. And in the process, we are going to drop off ApoC2 and ApoE from an HDL onto this chylomicron. So an HDL is going to kind of fly by and say, hey, take ApoC2 and take ApoE and throw it onto your chylomicron. And we're going to be like, all right, let's do it. So now we have a full chylomicron right here with our triglycerols still on the inside. And now we have ApoC2 bound to it, and we have ApoE bound to it, and we also still have ApoB48. Now, This chylomicron is going to come down through our capillaries right here. And when it gets into our capillaries, we are going to have this enzyme called lipoprotein lipase. And what lipoprotein lipase is going to do is it is going to get into our chylomicron and break up this triglycerol to release these fatty acids. And with a triglycerol, when we break down a triglycerol, we are going to release 3 fatty acids because it has the letters tri in the beginning of it, a triglycerol, 3 fatty acids and a glycerol backbone. So this lipoprotein lipase is going to release 3 fatty acids into adipocytes or tissues. For an example, adipose tissue. We are going to throw those fatty acids into the adipose tissue, but we still have a glycerol backbone left over, and this glycerol backbone is going to go to our liver. So nascent chylomicron comes through, runs into contact with an HDL. This HDL drops off ApoC2 and ApoE, and now we have this chylomicron come down. This chylomicron is acted upon by lipoprotein lipase. We release 3 fatty acids into adipose tissue, and we throw that glycerol backbone to the liver. And this chylomicron is going to come down here. And technically, at this point, it could be considered a chylomicron remnant. But what I really want you guys to know for exogenous sources and chylomicron specifically is we recycle and get rid of this ApoC2 to come back up here to this HDL. So we remove that ApoC2 back to the HDL, and now we are left with a chylomicron remnant. And we can see that we still have ApoE and ApoE48 bound to this chylomicron remnant. And what ApoE allows it to do is ApoE allows the remnant to bind to specific receptors on the liver where they're endocytosed. Kind of forgot what that word means, so I'm just going to say reabsorbed or recycled or whatever like that in the liver. But ApoE allows the chylomicron remnant to bind to receptors in the liver. Yeah. Yeah, okay. Yeah, I talked about everything in here. These notes, read through these notes, but I just basically just walked through that graph for you guys and it should be really good. So chylomicrons, that is how we kind of get rid of exogenous lipids in our body and put them into storage. So now we'll talk about very low density lipoproteins, VLDLs. And what we do know is these are for endogenous sources, lipids within our own body. So what we really need to know is the receptor on VLDL that ApoC2 and ApoE bind to is APO100. So this VLDL is going to be in, so when it's kind of in its form before it goes on all its transport, it's technically called a nascent or nascent, whatever you guys want to say, VLDL. Similar up here with the nascent chylomicron. Kind of same thing down here for VLDLs. So this VLDL right here is going to have this receptor called ApoB100 on it. And what ApoB100 is going to do is it's going to allow for ApoC2 and ApoE to bind to this VLDL. So this nascent VLDL is going to come through here with ApoB100, and it's going to allow ApoC2 and ApoE from an HDL to bind onto this VLDL. And this VLDL is basically going to go through all of the same steps we just talked about. It's going to come down here into the capillaries. It's going to have the protein or the enzyme lipoprotein lipase act upon it. We're going to release 3 fatty acids into, triglycerol tag. It's going to release 3 fatty acids into adipose tissue. We're going to get rid of that glycerol backbone, send it to the liver. But this is where it gets different. Now, after we get rid of that glycerol backbone or kind of break down that triglycerol, this VLDL is going to turn into an IDL. This IDL is called an intermediate density lipoprotein. This IDL is going to release ApoC2 and ApoE back to HDL, a little bit different because if you guys remember the last one in the chylomicron, we only released ApoC2, but in VLDLs, we release both ApoC2 and ApoE both back to HDL. And after it releases, this IDL turns into an LDL, which is low density lipoprotein. And we can see that we still have ApoB100 bound to it. And this ApoB100 allows it to bind to extra peripheral tissues. ApoB100 allows it to bind to peripheral tissues. Here's kind of the DNA change I was talking about. So high levels of intracellular cholesterol prevents the synthesis of ApoB100 receptors, which causes an LDL accumulation because it really just can't get back. Also too, like this, I forgot to say, this LDL can be brought back to brought back to the liver and recycled and reused if it wanted to. So it has two pathways: extrahepatic tissue or liver cells. Actually, those are both the same thing. All right, I'm all confused now. JK. So basically, when we have too much intracellular cholesterol, changes to the DNA can no longer make LDL receptors, and it also inhibits HMG-CoA reductase. So we kind of flash back up here. This is where that DNA issue happened when we have too much. Sorry, I just pointed at it with myself. This little thing right here, this little DNA change can occur. Oh crap, too far. And basically, this little DNA change inhibits HMG-CoA reductase, so no new cholesterol can be made within the cell. Yeah, all right. High-density lipoproteins, HDL, a good cholesterol because it converts cholesterol to cholesterol esters. If we remember esters are storage forms of things, so it allows us to convert cholesterol into a storage form of cholesterol. And it uses the enzyme LCAT. Lock this into your brain. Cholesterol to cholesterol ester uses lecithin cholesterol acyl transferase, LCAT, which catalyzes the reaction, lecithin cholesterol, blah, blah, blah. Cholesterol ester. I don't know how big of a detail that is right there, but hey, knowledge is power. HDL also produces the receptor protein ApoC2, which is needed in the membranes of VLDL and chylomicrons. We know that. This stuff right here, I don't know, man. Like, this is, you guys got it. You guys are smart. You guys can figure this out. This stuff right here. Little chart. All right. Lipolysis. Oh, dude, we only have like 7 more pages. All right. And we get to skip a lot of stuff too because I'm not talking about it. Well, I don't think it's that important, but you guys can read it on your own. So lipolysis, fat mobilization. Main purpose is to transport fatty acids from adipocytes to hepatocytes. Whoa, releasing fatty acids. We're releasing the fatty acids that we put in the fat back into the hepatocytes to use for energy, man. So it's stimulated by glucagon by states of hypoglycemia or epinephrine during fight or flight responses. We've already talked about that. I hope you guys are familiar with that. It is inhibited by insulin in response to hyperglycemia, which we know insulin is an anabolic pathway. So we're not breaking down stuff to use for energy when we're using insulin because insulin is anabolic. We're making stuff. Insulin would be more in the fact that we are putting those fatty acids into the adipocytes. And when glucagon and epinephrine are present, that is a catabolic pathway. So that's when we break down those adipose tissues to release energy back into our system, or we go through the process of lipolysis. So here's the steps. Adenocyclase uses 1 ATP to make CAMP. Whoa, we've already talked about CAMP. CAMP, what does it activate? It activates protein kinase A. We've already talked about that. Protein kinase A activates intracellular lipase. This is where it gets a little different. Intracellular lipase converts TAGs to DAGs to fatty acids. So basically we're taking triglycerols, putting them into diglycerols, and then we're converting those diglycerols into individual fatty acids. These fatty acids can leave the adipocytes and enter the blood now. And once these fatty acids are in the blood, they combine with albumin, which converts a fatty acid into a free fatty acid. Look at that differentiation there. A fatty acid is not a free fatty acid until it combines with albumin in the blood. And albumin makes this fatty acid soluble. And also, free fatty acids, fatty acid combined with albumin, can be transported in the blood. A fatty acid cannot be transported in the blood, but a free fatty acid, hey, that's free to go wherever, man. He's living large. So these free fatty acids can either go to hepatocytes, cardiac or skeletal muscle cells, or renal cells. Free fatty acids are eventually made into CO2 or ketone bodies, and specifically ketone bodies only in the liver. And what's even cooler about that is ketone bodies are used for energy for central nervous system neurons, and I believe red blood cells as well. But I'll have to fact check myself on that. So like right here, only ketones can go to the central nervous system to provide energy. And central nervous system neurons and red blood cells are the only cells that cannot oxidize fatty acids into CO2 for fuel. So we'll just kind of walk through this little chart right here. And this is basically just going to cover all of the steps we just talked about right here. We have ATP, acts upon and gets acted upon by adenocyclase. We cleave CAMP. CAMP turns into protein kinase A. What protein kinase A is going to do is it is going to activate intracellular lipase right here. This intracellular lipase is going to turn, we're just going to do that. This intracellular lipase is going to turn a triglycerol into a DAG and then into a fatty acid. This fatty acid is then going to get into the blood, or right when it's about to get into the blood, it combines with albumin to turn into a free fatty acid, because then it is free. It is free to go wherever. So it can go to skeletal muscle and we produce CO2. It can go to renal cells and we can produce CO2. And it can go to cardiac cells and we can produce CO2. But it can also go to the liver with hepatocytes. And these hepatocytes can create ketone bodies, and these ketone bodies can go to the central nervous system to create energy. And this whole system is activated during states of hypoglycemia. Yeah, I'm not an artist. I'm not going to try and do that. Yeah, okay. So yeah, that's lipolysis right there. And remember, fatty acids inhibit the production of pyruvate via glucose glycolysis. Yeah. Yep. All right, right here. We've already kind of talked about this earlier with gluconeogenesis, on how an overaccumulation of acetyl-CoAs can inhibit the PDH system and cause pyruvate to be turned into oxaloacetate to go through gluconeogenesis. So when we have these acetyl-CoAs, an increase in ketone bodies and free fatty acids inhibit glycolysis, since they cause an increase in acetyl-CoA production, and this inhibits the PDH system. This is basically a concept we've already talked about, but it never hurts to go back over it. This stuff in here, hey, knowledge is power. Go for it. Starvation, you guys can go back. You guys can go back and look at the starvation chart that we talked about, like insulin and glucagon, on your own time. I'm not jumping back there. All right. So now we talk about amino acid biosynthesis, and you guys are probably sitting here like, holy cow, I'm sick of hearing this guy talk. There's so much stuff here. I hate biochemistry, da 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 da. Well, hey, don't say that. Biochemistry's fun. So, amino acid biosynthesis. Amino acid biosynthesis, we really don't need to know all of this. What we really need to know are these essential amino acids, these 10. And we have a little acronym, Private Tim Hall. So we have phenylalanine for P, valine for V, threonine for T, tryptophan for T, isoleucine for I, methionine for M. I'm going to do all of them. Histidine for H, arginine for A, leucine for L, and lysine for the second L. And what I just want you guys to know is mainly these guys right here. If you guys can memorize all these, and he's like, what's an essential amino acid? And you see a couple of these up there, and you're like, well, that's not part of the acronym. Then hey, don't worry about it. But don't get confused, because we have an A down here, and we have like 3 A's right here. So be sure to memorize arginine and not mix it up with alanine aspartate or asparagine. Asparagine. I'm just going to call it asparagus. Not essential for infants. This is essential for infants, but not adults. I'd say that's a fun fact to know. These two non-essential amino acids depend on essential amino acids for production. Hey, memorize that on your own time. Memorize those 2 little fun facts, but I'd say know this for sure. All right, pyruvate alanine cycle explains how amino acids are mobilized to make glucose when fasting. Well, in healthy individuals, body protein remains constant because protein synthesis matches the rate of protein degradation. Dietary proteins supply the body with the necessary nutrients for tissue repair and amino acid availability. You guys can remember those statistics right there. But what's a cool fact to note is removal of just one essential amino acid in the diet causes a negative nitrogen balance, because we know a lot of these amino acids are helping transport nitrogen around our body. And amino acids have many destinations, which we kind of talked about earlier. You guys can memorize that in your own time. But during fasting, amino acids are mobilized from skeletal muscle to the liver, kidneys, or intestines so that they can be used in gluconeogenesis or ketogenesis. And we've actually talked about these 2 concepts already, and you guys just don't even know it. If you guys remember the Cori cycle and the urea cycle, these 2 amino acids have been seen before. So alanine right here must go to the liver to be used, and this is due to 2 reasons. Nitrogen removal as urea can only occur in the liver. And only the liver has the enzyme glucose 6-phosphatase, which is needed for gluconeogenesis to increase blood sugar. So I'll show you how alanine gets from skeletal muscle to liver cells. If I can remember where the Cori cycle was. A little flash from the past right here. This video is so gosh darn long. All right, here we go. Alanine, skeletal muscle. Cori cycle. We can see alanine leaves the skeletal muscle, gets to the liver, and when this alanine gets to the liver, we can go up through the urea cycle right here to cleave off that nitrogen, or we can go through a transamination reaction to be turned back into pyruvate. And this pyruvate will then go through gluconeogenesis all the way back to glucose, or alanine can go through the urea cycle right here in the liver. All right, all the way back down. And then glutamine must go to the kidneys and small intestine, moves to the kidney because to maintain the acid balance concentration of the blood by nonating a nitrogen to form ammonia. And I believe we've already talked about this too in the urea cycle. And I might be going crazy here and might be a fever dream, but where's the urea cycle at? Yeah, I mixed it up with glutamate. My bad. All right, fever dream. Oh, all right. Yeah, just know glutamine goes to the kidneys and small intestines. Failure of the pyruvate alanine cycle results in metabolic acidosis due to the formation of inability to dispose of ammonia as urea. And we talked about this last try in Biochem 1 with metabolic acidosis. And we know that we had compensations for metabolic acidosis either by hyperventilating to remove that CO2 or by urinating out NH4 plus. And this is kind of where that concept comes into play because we have an inability to remove that nitrogen or ammonia as the form of urea. And yeah. All right, we're just going to keep on cracking. I might be, I don't know. We're just going to keep on cracking away. So we have methane. Methane is a toxic, colorless, odorless, flammable material. This stuff, if you want to memorize it, do it. Methionine is a main methyl transporter that is formed during the decarboxylation of 5-methylfolate to folate. We're going to be getting into this. So in food, it is folic acid, vitamin B9. In food, it is labeled as folacin. Folic acid must be consumed in the diet since humans cannot synthesize it. Tetrahydrofolate is an active form of folic acid that is produced from removing 4 hydrogen. I don't care. All right. Realistically, what we need to know for this is 5,10-methylene THF turns into 5-methyl THF and it will eventually turn into folate. But we need homocysteine to react with 5-methyl THF to remove this methyl group right here, and is going to be using the enzyme methionine synthase. Homocysteine is going to react with 5-methyl THF to remove that methyl group at being acted upon by methionine synthase to create methionine. Homocysteine turns into methionine when a methyl group is added. Methionine is now going to react with norepinephrine. When it reacts with norepinephrine, it creates epinephrine, and we need the B vitamin B12 methylcobalamin. So, recap, 5,10-methene THF gets turned into 5-methyl THF. Homocysteine, with the help of methionine synthase, will react with 5-methyl THF to remove that methyl group and to turn 5-methyl THF into folate. Now, when homocysteine picks up that methyl group, we turn into methionine. Methionine will then drop that methyl group off to norepinephrine with the help of methylcobalamin B12, and norepinephrine will now turn into epinephrine, and we can use it for our fight or flight response. And then methionine will turn back into homocysteine after dropping off that methyl group, and it can just be reused for the cycle again. And then purines and pyrimidines required for folic acid synthesis. For purines, we need adenine and guanine, and for our pyrimidine or whatever, we need thymine. And the only way to make homocysteine in the body is to remove a methyl group from methionine. We just talked about that. Failure of this process above would result in a deficiency of methionine, an essential amino acid. And basically, if this all fails, we do not pick up that methyl group from methionine, and we can't transport methyls around our body. Methylcobalamin B12 is an enzyme derived from cobalamin that is that transfers a methyl group from methionine or epinephrine to form epinephrine. Um, We talk about a trap. There is a trap. Cobalamin, read this. Um, yeah, read it if you want. I don't care about this stuff. Do what you want with that. A B12 deficiency causes a folic acid trap because 5-methyl THF cannot turn into folate, and the reaction a 5,10 methionine or methylene THF to 5-methyl THF is irreversible. So basically, what this is saying, when we have a cobalamin deficiency or vitamin B12 deficiency, and we can't create methylcobalamin, basically what's happening is we do not have this anymore. We do not have vitamin B12 right here. So we cannot have this whole reaction to go. We cannot have this cycling of methyl groups right here. And as a result, we do not get homocysteine again to react with 5-methyl THF right here. So we have an overaccumulation of 5-methyl THF here, and it is not reversible. So we just keep on getting that huge accumulation right there. Yeah, and we need folate to produce nitrogen bases for purines and pyrimidines. So yeah, I kind of flew through that, but you guys can always rewind it. That's why I like making these videos, so I don't have to repeat myself a whole ton. So now we talk about purine synthesis. There are two purines, adenine and guanine. All purines have ribose 5-P backbones that is acquired through the pentose pathway. I hope you guys remember the pentose pathway because it's starting to reappear. Nitrogen is provided to the purine synthesis pathway from glutamate, glycine, and aspartate. Memorize that. Purine synthesis involves many steps, but starts with the phosphorylation of ribulose 5-P into 5-phosphoribosyl pyrophosphate, PRPP. PRPP, just memorize that, and it's accomplished with the help of phosphoribose pyrophosphate synthase, PRPPS. Oh man. Basically, what we need to know from this conjumble of words right here is ribulose 5-P is acted upon by the enzyme PRPPS to create PRPP. Boom, there we go. That's how we get ribulose 5-P or ribose 5-P to turn into PRPP. And then PRPP goes through 10 steps until it's formed into inosine monophosphate, aka inosinic acid. And this IMP right here is used to create adenine and guanine, which are our purines. So if we look right here, we have IMP, and we have two separate pathways we can go down. We can go down a pathway that throws on aspartate, and we do that. We use adenosylsuccinate synthase and use it in IMP reacts with this enzyme to come down here and make adenosylsuccinate, and then adenosylsuccinate will eventually get turned into adenine. But if we don't want to do that, we can throw on an H2O and use the enzyme IMP dehydrogenase, and we can basically turn it into guanine down here. So if you see the addition of an aspartate to IMP or IMP reacting with adenosylsuccinate synthase, it will be made into adenine. But if you see IMP being acted upon by IMP dehydrogenase or adding on an H2O, we know that is going to be made into guanine. And then right here, what I just talked about. And then high levels of GTP, GMP, and AMP inhibits PRPPS. So high levels of AMP, high levels of GTP, blah blah blah. It inhibits this enzyme and does not and prevents the conversion of ribose 5-P to PRPP, which then prevents the creation of inosinic acid or IMP. And then supply of adenine and guanine is determined by what is missing. Now we'll talk about pyrimidine synthase or synthesis. And I really don't know too much about this, but there are 3 pyrimidines: cytosine, thymine, and uracil. Folic acid is a precursor to thymine That actually arrow does not look good. That's what uric acid is made into, and this stuff, again, I really do not know a whole ton about the inner working details on what to know. If I had to remember anything, remember common bile synthase 2. Carbon, we'll call it CP, that is a product of the urea cycle, and just go back and take a peek at the urea cycle. I forgot exactly where it ties in, but just know that is where the carbonyl P comes from. And it leads to the formation of UMP. UMP is a shared precursor for cytosine, thymine, and uracil, and it's phase-determined by enzymes. Pick this apart if you guys want. I apologize. That is nothing that I cannot do that. So now we talk about uric acid. Uric acid is not urea. As purines decay, they are converted into uric acid, which is then excreted in urine. Adenine and guanine are both converted to xanthine, whatever. Memorize that. Just know A plus G goes to, actually A or G goes to X, which is an intermediate in the formation of uric acid. Xanthine oxidase is the enzyme that is used to convert hypoxanthine to xanthine to uric acid. Failure of the body to excrete uric acid results in hyperuricemia, which is gout. Gout is a form of arthritis that affects a lot of people, and I believe it happens quite a bit in the foot, because I remember seeing shows always talk about, like, oh, my gout's acting up, and I'm like, oh man, what does that mean? Well, now we know. Hyperuricemia is an increase in uric acid, which inhibits PRPPS. And if we remember, PRPPS is the enzyme that converts ribose 5P to PRPP, which then will eventually go to IMP, which will eventually lead to arginine and guanine. So that means if we have an increase in uric acid that we are not excreting, we have a decrease in PRPP because we do not have this enzyme, which means a decrease in IMP, which means a decrease in purine production. And gout mostly affects males. All right, so we made it to the end of this packet. So this packet, I don't know, the other packet, I don't know how many, if I sent this out to a whole ton of people, but we are just going to fly through this. This neurotransmitter stuff, pause the video, look at this stuff. It's kind of a review of neuroanatomy, so just read through this, get a good understanding of it. Iron metabolism, we talked about this a little bit last try with proline, with the creation of proline for collagen formation with the use of ascorbic acid and vitamin C, and how it oxidizes and reduces iron. Iron component of hemoglobin, Fe2+, synthesis of collagen, ascorbic acid, vitamin C re-reduces iron back into its normal state from Fe3+ into Fe2+, so it can donate something, I believe, in oxygen to proline for collagen formation. Fe2+ is more soluble than Fe3+. All right, so here's the definitions we need to know. We need to know ferritin, which is the storage form of iron. It's an iron-containing polypeptide. Iron is stored in the liver. We need to know transferrin, which transports protein, which is a transport protein for iron. And then hemisterin, it forms when there's excess iron. So use that, and it functions to destroy liver cells and relieve excess. So if he talks about destruction of liver cells with iron, you are looking for hemosiderin. Uptake of iron by mucosal cells is regulated by the amount of apoferritin and apoferritin. So basically what's going to happen is we eat iron in our diet, Fe3+. It gets into our intestinal lumen where it becomes reduced into Fe2+. This Fe2+, because it's a more soluble form, it can pass into our mucosal cell. Iron 2+ is going to react with apoferritin, which is a precursor to ferritin. So when iron reacts with apoferritin, it turns into ferritin, and ferritin is our storage form of iron in our mucosal cell. Now, if we want to transfer it and use it for red blood cell synthesis or bring it to the liver, we will oxidize it. We will oxidize this iron and get it out of its storage form. So we'll take it from Fe2+ and create it back into Fe3+. This Fe3+ will now combine with apotransferrin, and it will turn into transferrin, which is the transport mechanism. Transport mechanism to get iron to the liver. So we can see that we still have an Fe3+ here with transferrin. So when it goes to the liver, it becomes reduced again. So now we will have ferritin We have ferritin in the liver, which is the storage form of iron in the Fe2 plus form, or we can also take it and put it into bone marrow for red blood cell synthesis. Yep, rewind that if you didn't get that. That stuff, hemolysis occurs in the spleen, breakdown of red blood cells. Lifetime is 120 days. They get broken into globulin, which is recycled for amino acids. Then we have the heme group. This heme group is made into bilirubin, and this bilirubin is excreted in the feces. Gone. Only way to lose iron is fecal excretion. And so we'll talk about jaundice here. Jaundice is due to a non-soluble accumulation of bilirubin in blood plasma in infants, affects the skin, whites of eyes, etc. Clear sign of liver cirrhosis and liver failure because these bilirubin is just destroying some of these liver cells. And then I believe if you expose infants to sunlight or blue light, it helps break down this bilirubin and kind of reverse the effects of jaundice. Okay, guys, this is it. This is the home stretch, and then you are done hearing from me until the foreseeable future. So, alcohol metabolism. We will talk about alcohol metabolism here because ETOH, ethyl alcohol, is a 2-carbon alcohol and it is water soluble and goes into your blood fast. I like beer. I think you guys like beer. Or what are those? Happy Dads. Honestly, I'm trying to think of one of those seltzers. High Noons. That's what I was thinking of. Okay, so catabolism of ETOH. We're going to have two forms. We're going to have ADH, which is alcohol dehydrogenase, which is found in the cytoplasm. Then we're going to have ALDL, aldehyde dehydrogenase, which is found in the mitochondria. And both of these are enzymes. So, cytoplasm, mitochondria. So, ethyl alcohol is going to be reacting with ADH, or alcohol dehydrogenase, to break it into acetaldehyde. And we are going to use an NADH to come and pick up a hydrogen off of that because it is a dehydrogenase. We are removing a hydrogen from ethyl alcohol. So once we remove that ethyl alcohol, this NADH is going to come down here. It's when it picks up that hydrogen, that NADH is going to come down here, react with pyruvate, and we're going to drop off that hydrogen to convert pyruvate into lactate. And then we'll be left with acetaldehyde. And this acetaldehyde, what it's going to do is it's going to react with aldehyde dehydrogenase. And yet again, it's dehydrogenase. We are removing a hydrogen from this ethyl alcohol. And we are taking this NADH because we picked up the hydrogen from here. We are taking that NADH, reacting it with oxaloacetate to convert it back into malate. Because if we remember, if we have malate and we remove a hydrogen from malate, we turn it into oxaloacetate. So if we add a hydrogen to oxaloacetate, we turn it back into malate. Yeah, so basically, when you drink alcohol, you are using up NAD plus storages within your body. And we'll talk about why you get hangovers in a second. But this system right here is using up NADHs or NAD pluses that you need for other mechanisms within your body. So alcohol catabolism occurs in the liver, and this is where a lot of these issues come from. So acetate will result in ketone bodies being made, because our final product right here is acetate, which is a ketone body, and we know ketone body formation occurs in the liver. So if we have a ton of ketone bodies, that puts us in a primary alkali deficient because we are using, when we break down these ketone bodies, we are using bicarbonate ions, which are removing basic components from our blood, which causes our blood pH to increase, putting us in a primary alkali deficient because we're using up these basic components. And if that goes on for a long time, it puts us in metabolic acidosis. And metabolic acidosis, again, is a chronic condition of high blood or low blood pH, acidic pH within our blood. And if we remember from Biochem 1, when we are in metabolic acidosis, the only way to compensate for that is to vomit, to get rid of these acidic components. Now, secondly, the amount of NAD plus decreases within our body. This results in several problems. We have lactate acidosis. We need NAD plus to convert lactate back to pyruvate, but we can't because we're using up these NAD pluses, converting pyruvate into lactate. And this increases the lactate concentration, which leads to an increase of hydrogen concentration in the blood, which will lead to an acidotic state because acids or hydrogens are an acidic component and lower our pH. The inhibition of gluconeogenesis, the use of TCA components and pyruvate, because we can see we are using oxaloacetate and pyruvate, which are both gluconeogenic components. It decreases the number of carbohydrate precursors, which is limiting our glucose production. So if we have a low drop in blood sugar or a drop in blood sugar to a low amount and we are drinking, we are taking away components that we could be using to increase our blood plasma levels. And it results in hypoglycemic effect because we can't efficiently create glucose to put back into our blood. And then we inhibit beta oxidation due to the decreased amount of NAD+. I really don't care too much about this stuff. Do with what you guys want. And hangovers, they are due to acidosis, hypoglycemia, and dehydration. So acidosis coming from the ketone body formation, hypoglycemia coming from the inhibition of or initiation or the inhibition of gluconeogenic precursors. And then we also have dehydration, which is just us using water components for other things, and water recruitment is water reabsorption is decreased while we are drinking. So if you don't want to get hungover before you go to bed, eat a Jimmy John sub, have some liquid IV to stay hydrated, and vomiting. I don't know. You guys do what you need to do, man. I don't know. If diabetic, you're at serious risk because you already have a hard time regulating your blood plasma levels. Long-term problems, cirrhosis, I'll let you guys finish that off. But yeah, that's it. Oh man, that's a great way to end it. Thank you guys. I had a lot of fun tutoring everyone over these past couple of tries, and I'm really hoping I helped you guys out with biochem. So, if you're gonna do something, be the best at what you do. I'll leave those words of wisdom with you guys, and yeah, keep it crack-a-lackin'. Have a good day.